The test was conducted to investigate the efficiency of using luteinizing hormone-releasing hormone, LHRH, and human chorionic gonadotrophin, HCG, to induce spermination and ovulation in the southern Corroboree frog, Australia's most critically endangered frog species. This provided an opportunity to test whether hormonally induced sperm and oocytes could be used to generate embryos via in-vitro fertilisation, IVF. Male frogs were administered a single dose of either HCG or LHRH, while female frogs received a priming and ovulatory dose of LHRH. The doses were administered via subcutaneous injection and based on successes in other endurance. Spermination responses were evaluated at different intervals for 72 hours, and sperm number and viability quantified using fluorescent microscopy. Ovulation responses were evaluated by stripping females every 12 hours for 5 days. Once gametes were obtained, IVF was attempted by combining spermic urine with oocytes. Embryonic development was checked every 6 to 12 hours for 7 days. Of the male frogs, approximately 80% released sperm. On average, LHRH-treated males released a significantly higher concentration and total number of live sperm, indicating its superiority over HCG. This is illustrated in Figure 3, where the mean sperm concentration is significantly higher in LHRH than in HCG. Figure 4 shows that the mean total proportion of live sperm released was almost 10% higher in males treated with LHRH than with HCG. Although hormonal treatment was successful, males released significantly lower amounts of sperm than previously reported for endurance. This may be because the doses did not promote optimal response, therefore testing across a range of doses may be necessary. In female frogs, approximately 30% of individuals released eggs. This proves that LHRH was successful in stimulating ovulation. However, the percentage of females responding was low. Post-experimental inspection of unresponsive females revealed that individuals were carrying immature oocytes, which may be the reason they failed to ovulate. The average clutch size recorded during this experiment was 15, which is small for this species. This can be seen in Table 2. This may suggest that LHRH lacks potency in females as was suggested for males. IVF resulted in 55% of eggs being fertilised. This variable level of success may have occurred because sperm concentrations were suboptimal or the fertilisation medium was inappropriate. Further work will be needed to identify the primary cause of low fertilisation success. Of greater concern is that all embryos failed prior to gastrulation. This may have been linked to genetic incompatibility as the frogs were sourced from mixed populations. Similar studies conducted by R.K. Brown and J. Surratt also produced low levels of embryo survival, and they suggested that hormonal induction impaired gamete viability. Hormone treatment successfully induced spermination and ovulation in the southern Corroboree frog, but refinement of gamete induction and IVF techniques will be required before assisted reproductive technology protocols can be used to augment captive breeding in this critically endangered species.